Since I entered to the field of forensic science in the early 90s, unfortunately because of identification of war victims from the mass graves, it was always idea to have outstanding group of the forensic science at one place and cover everything from the crime scene investigation till the court. So I was lucky with Professor Moses Schoenfeld from George Washington University to have all these authors together. And basically what we are doing with this book, we are covering some basic genetics, forensic statistics, analysis of the mitochondrial DNA, genomic DNA, Y chromosome, X chromosome, and then we are a little bit entering into the different procedures, how to extract DNA, how to amplify, how to use different systems. We also cover chapters of the animal and the plant DNA. Then we are also discussing about different new technology like microarray technology and next generation sequencing, and getting very, very deep in some very, I would say, concrete cases such as 9/11 case, identification of victims from such a difficult, horrible terroristic attack to the United States of America. And we are moving a little bit further and covering identification of skeletal remains, what we have case in Croatia and Bosnia. Moving further and basically, I would say, talking about completely new discipline, what we call it's forensic phenotyping. We are trying to describe how the person look like according to some genetical markers. And finally, we are entering to the arena of what we call the molecular autopsy. So we are trying to find the different biomarkers that will help us to find the cause of the disease and eventually cause of the death that usually not able to see by classical or typical forensic examination. And at the end of the book, we are entering to the court and trying to describe which kind of skills forensic experts should have once when he's giving testimony on the court, discussing different ethical issues and horrible problem that we are all facing today. It's on the human trafficking. So this book is really covering all aspects of the forensic DNA work, and I was so proud to have such outstanding group of the people together in this book. It took us more than 2.5 years, and as an editor I have a very difficult and not very pleasant task to push the people. So I was really forcing people to be on time. But again, you know, most of us are really, really friends, people with incredible reputation, and once again I would like to thank them for their time and dedication. I would say the book was extremely well received by the students, by experts, by police officers, by military people, by Secret Service people. So basically everyone who is involved in forensic science. And I just learned that the new edition on the Croatian will be published. In 1993 I did with my colleagues from Split University the first DNA forensic laboratory in that part of the Europe. And according to the information that we had, in our lab actually happened the first identification of skeletal remains in the Europe ever, particularly working with the skeletal remains from such huge catastrophes as it is war. So we do have right now laboratory in Split as a part of the clinical hospital Split, and we do have one private forensic laboratory in Zagreb called Genos, outstanding lab with, I would say at this moment, 12 PhDs running different program forensics, including paternity cases, some criminal cases, but also new technology that forensic field is entering in, but also covering some clinical work like molecular diagnostics and other things. I'm a geneticist, also professor of the pediatrics, so I'm combining forensic genetics and also pediatric genetics, trying to understand basically, you know, on early 90s, I was studying, my PhD came from University of Connecticut. We tried to describe and to understand a disease called brittle bone disease. So I spent many years working with the kids with this particular disease. And I would say in 1994, 1995, we were able to describe particular mutation that was responsible for this disease. So we were able to find the mechanism why some kids getting severe, why some kids are getting moderate type of the disease. And later on we continue with the different work and entering to completely, and I would say so excited field of the medicine called regenerative medicine or personalized medicine. So what we are trying to do, we are trying to develop the very particular therapy for every single person that was depending on his genetic profile. So that's what is our goal in the future. For example, what we know today, that each of us having completely different metabolism of the certain drugs. And all this depends on the certain polymorphisms or the mutations on the gene that are particularly important to produce the enzyme which are going eventually to metabolize the drug. For example, 5 milligrams of the drug can help somebody and at the same time can hurt badly somebody because the metabolism of the drug is completely different. So what we are trying to do, we are trying to prevent patients from so-called adverse drug reactions. So that's my particular right now interest, working on the genetic and the clinical work. What we are facing today, we are facing so many problems because of the drug interactions. Unfortunately, so many people receiving more than 2 or 3 drugs, and it's really not much known what is the interaction between drugs. We know a little bit about that, but you know, it was a very, very interesting article published in early 90s in the JAMA, showing you that almost, I would say, 120,000 people are dying each year, the people who are hospitalized because of different adverse drug reactions. By understanding pharmacogenetics, we are able to prevent all these people from dying from this particular, you know, drug adverse reaction. So it's incredible new field and new area that is opening. And, you know, it's kind of combination, as I just told you, clinical work, but also forensic work. Because you know, it's very well known that in so many cases incredible negative reactions on different drugs can happen. And indeed, forensic expert is one who is going to give explanation why this happened. I'm very much interested on the cellular therapy, very much interested in the gene therapy, and as a matter of the fact, in my hospital in Croatia, we recently, for the first time in the Southeast Europe, apply new technology. It's application of mesenchymal stem cells in the knee of the patients who heavily suffering from the cartilage damage, with one idea: to stop inflammation and eventually to stimulate some of these stem cells to produce the cartilage. So we just enter to the study and we'll be waiting for the next year, doing different, you know, clinical examinations, doing MRI, and doing different technology where we're able to measure, you know, very important markers of the cartilage cells, what we call chondrocytes, proteoglycans and collagen type 2, and eventually we will be able to show that some significant changes happening after application of the mesenchymal stem cells. Basically, you know, as you know, education is so important for us. So I was so proud that American Academy of Forensic Science, you know, thanks to you and all other friends, last year signed the contract with the International Society for Applied Biological Sciences that I'm in charge of currently. So right now, we are with the jointly forces organizing different conferences, particularly in Croatia. So I'm very pleased to announce the next one will be on 2017, June 19th to June 24th. And at this moment we have confirmation of 5 Nobel Prize laureates who are coming and who are going to be part of the conference. I'm so proud that American Academy of Forensic Science is joining us in that particularly important project. And usually we are having more than 500 participants from 60 different countries. So the value of the collaboration between International Society for Applied Biological Sciences and American Academy of Forensic Sciences for the field of the forensic science, it's incredible. And I'm so proud to be part of all of that.